view of the environment. And I'm going to really try to discuss how our microbiome is basically a continuum with the microbes that are on the planet. So to me, environmental health is the crosstalk or communication between our microbes and the rest of the microbes on the planet. That is, I think, probably the most liberal kind of view of environmental health that you might hear today. So my talk is going to have 4 parts to it, basically 3 parts. What is the human microbiome? That was what was charged to me. But I also want to go into how does it form, because that's tied to the environmental question. And then more explicitly, what role does the environment play in the microbiome? And then I'll try to wrap it up. Ever since Anton von Leeuwenhoek, right, he's the one who was this person who developed the first microscope, you know, we knew that the human body was full of microbes. He took, you know, dental scrapings and so on and found these lovely little animalcules. So it's taken, though, unfortunately, about 4 centuries before we could come back and revisit the question of the microbes in and on the human body that were not just strictly about pathogens. So, though, and I'll just read this to you, though we as the public think of diseases being caused by microbes, and I offer a couple examples, in fact, the majority of microbes that we interact with do not cause disease. Okay? Here are 4 or 5 examples. They're certainly important in food production. Look at some of the foods like bread, cheese, yogurt, and so on that microbes are important in producing. They're certainly key to soil. So the majority of microbes that we interact with, though, don't cause disease. Okay, I already mentioned food production. Certainly, we wouldn't have soils without microbes. Nutrient regeneration, the issue here at the table, pollutants and toxin degradation and recycling. Certainly, oxygen production. Something like of every 2 breaths you take, half of the breaths come from microbes in the ocean. And then to the point, today, they play a role in human health. So let me just kind of describe what the human microbiome is. Okay, the human microbiome, not microbiota, the human microbiome is really a term to describe the full complement of the microbes and their genes. That's key, and their genes in and on the human body. These are sort of like, you know, bullet points, take-home points. There are 10 times more microbial cells than human cells. You heard these kind of things in the lay literature. The human microbiome, and now I'm talking about the whole human microbiome. Certainly the key microbial organ is the gut microbiome, but all body sites have its own unique flora, or microbiome. They're on the order of around roughly 1,000 species of microbes. More importantly, there are about roughly 3 million microbial genes. Now, if you contrast that against the human genome, which is 23,000, you can see how that genetic signal is really, really important. To give you sort of some take-homes, we're carrying around 3 pounds of microbes in our gut. Something like half or so of our stool dry mass is microbial mass, and that's important because you can now get a picture of how fast the turnover rate is of the microbial community in our gut. And then, as I said a moment ago, each body site and the human microbiome actually is addressing 5 major body sites. The gut, the skin, the oral, the nasal, and the urogenital all have unique and characteristic communities. So I'm not going to go into the roles. I'm not going to address them per se, but these are obviously 4 key roles that we can't live without. They protect us from opportunistic pathogens. They help develop and maintain our immune system. Certainly without our gut microbes, we couldn't actually digest a lot of the foods that we consume. They produce energy because the final end product of gut digestion and fermentation are short-chain fatty acids, which is what our cells need for energy. And then finally, they produce all kinds of beneficial compounds and anti-inflammatories, antibacterials, vitamins, things like that. So let me walk you through, though, the kind of the natural history of the microbiome, because the microbiome, and I want to say this a few times during my presentation here, you know, we inherit our genome, the eukaryotic genome, us, but we actually, each generation, acquire the human microbiome. So that's where the environment signal is really, really important. The human microbiota are acquired anew from the environment each generation. Okay, you might think it's a stochastic process, but it's not. because here are data, and this is pulled from, I try to reference each source that I pulled data from, so if you want to know, I can give you more details about the citations. In some cases, I'll be presenting unpublished data, so I can give you the contact name for that individual. Infants acquire their initial microbes from the mother during sort of normal vaginal birth, okay? And here is a diagram, a PCA plot in color. The mother's body habitat microbial microbes are in bolder colors. Oral is in green, vaginal is in red, and skin is in blue. In the case of the baby, here are the metagenomes from baby delivered vaginally versus babies delivered by cesarean. And what you should see right away, and I circled it in red, is that the initial inoculum is actually coming from the vagina. And it's actually even more amazing than that. During the 9-month gestation period, the mother's vaginal microbiome kind of coalesces and develops into this inoculum ready for the baby to be born. Okay? However, babies are kind of a microbe magnet. And the reason I want to point that out is even when they're born cesarean, okay, they still pick up, oh, pardon me, I hit the wrong button, still pick up microbes. So here's the example where the baby was born by cesarean, and the main inoculum for that baby is from the skin. Now, the skin is not necessarily the mother's skin. It could be actually anybody who's handling the child. So that's sort of point number one about the development of the microbiome. Point number two is actually, though that first, so the inoculum comes from the mother or wherever the main source of microbes it's exposed to, and because it's a microbe magnet, it's a kind of a bioreactor, okay, over the first few weeks to years. And here's another set of figures from another paper, Palmer et al. 2007. Let me just orient you for a minute. This is actually on log scale on the y-axis in each one of these little panels, and on the x-axis is time, and we're talking about over days. So that's not quite a year's worth of data there. And what I want to point out is that just looking at total abundances, bacterial abundances increased by about 6 orders of magnitude in the child. You can see that these are different children, different babies, and so actually it's sort of a dynamic process. A lot goes on, and as the system kind of adjusts, and the microbes are talking to the gut, and the gut's talking to the microbes, and that kind of thing, right? So, but we will see that within about the first year of life, so this is data from the same study, days, and this is correlation to the adult microbiome, that basically, and this is my red arrow, not theirs, basically you start seeing this trending of the composition of the community coming together and becoming more adult-like. So there's actually some kind of, process that happens to allow these microbiomes to establish. In the adult, basically, as I mentioned before, the microbiome composition is unique to each body site, but what's interesting, very interesting, is apparently it's not related to gender. It's not, I mean, even between people, there's not much difference, and over time, there's not much change. But what we do see, and here are the data to support it, that these are habitats or body sites, okay, hair, oral cavity, gut, skin, and nostril, and I've just forgotten what EAC stands for. External auditory canal. Thank you. Thank you, thank you. You can see earwax. Okay, earwax in red. You can see basically that this PCA plot shows that the microbial composition clustered according to body site and not any of the other more seem to be kind of intuitive parameters. Okay, so microbes are forming their own habitat there at each body site. But there's even yet more information about the kind of natural history or dynamics of the microbiome through the lifetime of an individual. And that is that it's fairly stable through time until you start getting older. So this is the only really good elder study that I have, and it's from the Irish. It's called the ElderMet Project, and I've got it cited there. And what they showed here is the elders in their study against other recently published microbiome studies. And you will see, no matter if you're looking at whatever level of taxa, it doesn't matter, phylum, genus, or at the cluster level, is that the microbiome composition is actually quite different in elderly, and their definition of elderly was individuals older than 65 years old. Okay? And I think, if I remember correctly, it was like 65 to 90-ish, something like that. So the gut microbiome actually does change from sort of midlife into elderly adults, and apparently they have, I would call, a unique microbiome. So let me try to now go way out in terms of the environment and the human microbiome. And these are going to be data that aren't published yet, but I think it's probably part of the thinking that needs to be included in this conversation. So just to remind you, the human genome is inherited, and the human microbiome is acquired from the environment. So how does the environment play a role? This is from Eric Alm's lab at MIT, and he just recently presented at a at a meeting in Vancouver, the International Human Microbiome Project. And let me walk you through this because it's actually kind of a complicated figure, but it captures a lot of really important information. I want you to consider that, so one of the reasons why microbes are so successful on this planet is they don't limit their gene exchange to sex. They promiscuously share genes across all kinds of habitats and under all kinds of conditions. I mean, the best example we can offer is antibiotic resistance, right? We actually conducted an unintended experiment through the use of antibiotics, and now there's all kinds of antibiotic-resistant microbes, and that was done through horizontal gene exchange. So horizontal gene exchange, kind of a general definition, would be gene exchange in the absence of any kind of sex. And in fact, microbes do it very, very well, and they do it a lot. So horizontal gene transfer, let me just read you the title, is more frequent in the human microbiome than other metagenomes. The way Eric approached this, I don't know the computational tools that he used, but he compared metagenomes available in the public databases, and through some computational analysis tried to understand how much horizontal gene transfer there was occurring in different kinds of microbial habitats. Okay, so let me walk you through this heat map. These are human body metagenomes. These are non-human metagenomes. So you can see there's marine and soil samples. And I want to break it down over here. He also included in the soil samples sort of natural environments and then foods or farm soils. So that's kind of important. You can see, just looking at without even walking more through this diagram, that the hottest or the highest rates of horizontal gene transfer are occurring, compared to any other metagenome, is occurring in the human microbiome and also occurring in farm animals. So horizontal gene transfer rates are even higher among bacteria from the same body site. That's where you see the really red bars. And the second panel here, farm animals are the most frequent non-human source for gene exchange with human-associated microbes. So that's two take-home messages. There's lots of horizontal gene transfer occurring in the microbiome, and the source of genes, the most important source of genes, is coming from farm animals.
Is that thought to be because we eat farm animals, or is that thought to be because there is a certain subset of the population that lives close to them?
If I understand Eric's results, it's actually because we eat farm animals. Although you can't separate that, yeah. But it's because many of us are carnivores.
Because we feed farm animals antibiotics.
Yes.
Just going to say that the most important driver and the most important use of antibiotics in most countries, especially this one, is as feeds.
Subtherapeutic levels
of feeds. So
right.
You should expect the highest rates of HGT to be occurring in livestock, animals, and in farms. This should be broken down a bit more to be explanatory. In the animals that are receiving these feeds, because we already know that's driving not only evolutionary pressure, but it also increases the rate of horizontal gene transfer.
In general, not just for antibiotic resistance genes.
I don't know if we know that in general.
So-
I can't say that. I haven't seen any studies that have been apart from at least looking at
Oh, I guess I
cassettes
didn't
and
think
plasmids that include
Yeah.
resistance genes. I don't know that.
So you're absolutely correct, Ellen, but I think also the way that Eric approached this analysis is he used some kind of independent metric for looking at horizontal gene transfer. So I don't actually know the computational approach he used, but it was my understanding from him. I'm sorry, could you speak in the microphone again?
Sorry, it's the same that Amanoff has used in several recent publications.
Okay, thanks. And then the other take-home message from Eric's lab is that ecology trumps phylogeny and geography as the most important factor that's driving horizontal gene transfer rates. So what I want to try to walk you through here is on the left panel, here it is. On the left panel, okay, here's a 16S distance and horizontal gene transfer rates. And he plotted the horizontal gene transfer rates of different habitats or ecologies of the microbes. And you can see that the rates of horizontal gene transfer of microbes that share the same ecology, whether it's physical parameters or nutrient requirements or whatever, is much, much higher than microbes that have different ecologies. And in fact, even from the same, there's not much difference whether they come from the same continent or a different continent. So you think proximity would play a role, but it's not proximity, it's ecology that drives horizontal gene transfer rates. So the dotted line shows horizontal gene transfer amongst most distantly related taxa is more common with shared ecology than horizontal gene transfer amongst closely related bacteria from different origins. Kind of counterintuitive. So one more thing I want to try to introduce here is, especially if you come from a microbiological perspective, we've been dealing with human pathogens sort of in the absence of their environmental context. So this is a very interesting study. This is out of Volker Meyer's lab. He's at the Argonne National Lab and also University of Chicago. Again, unpublished data. What they did here is they also went into the public database and pulled out all metagenomes, and they blasted the NIAID's priority pathogen genera against that. Okay? And what they found is that all these different environments, marine, animal, and microbial mats, and so on, and I think these were not well characterized, showed very high presence of bacterial genera that has human pathogen members. I'm not saying these are actually the pathogens, but if you blast the priority pathogen list against these metagenomes, you see lots and lots of hits. And some environments have more, I'm going to use this term loosely, I don't mean to be too cavalier here, but pathogen diversity than other environments. Example would be, if you look at the marine environment, there's a lot of hits against NIAID's priority pathogen list, whereas in some environments, like here, microbial mats, there were very few. So the point I'm trying to make here is there's no, well, we have to start thinking of the human and the environment as kind of a continuum, because there appear to be perhaps reservoirs of either pathogens or pathogen-like properties in all kinds of environments that are outside of the human. And finally, I want to close with one last thing. This is not my idea. This is Marty Blazer. He's been sort of discussing this idea for some years now. It's considered that, I think he calls it the disappearing microbiome or disappearing microbiota hypothesis. And the idea there is our own common practices in kind of developed countries are actually having an impact on the kinds of microbes that, remember, these are acquired new every generation, the kinds of microbes that the newborn is exposed to. And basically it boils down to, let me look at the left-hand panel, contemporary practices like sanitation, clean water, bathing, antibiotic use, cesarean birth, formula feeding, and mercury amalgams in our fillings are all playing a role in kind of killing off or removing or reducing our exposures to typical floras that we were constantly exposed to over the evolution of the human species. And because each generation has potentially a more and more impoverished microbiota, then the idea here, and that's what this stepwise diagram is supposed to show, that as each generation passes, we have fewer and fewer sort of the full, rich inoculum that the newborn should be exposed to. So that's another kind of piece of information that I think is part of this conversation.
Excuse me, you could have a different interpretation,
Okay.
which is that most
Okay.
of those experiences would be associated with resistance. And therefore, what we're doing is, as Wright would say, enriching the resistome. And the microbes have a very, they've developed billions of years of handling these kinds of insults that we or nature delivers. So I'm not sure that I would, to exposures to chlorine, certainly mercury, antibiotics, absolutely, nutrients, bacteria can cope with that. So I don't think we would necessarily be depleting the microbiome.
Okay. Thank you.
So, Lita,
Yes.
So actually I'll come to the defense of that and actually say that if you look at a single microbe, you can clearly demonstrate the ability of a single organism to adapt. But when you look at polymicrobial communities, in the time that it takes for one organism to adapt, another fills its niche, and then you get competitive exclusion, and out it goes in a situation where you've got metabolic influences on it. So you can see community dynamic changes that may or may, and if that community is then passed from generation to generation, it's not that that disappearing microbe can't ever come back. It's just as you pass that along, you've lost it out of your, inoculum as you move through. So that's the theory, behind that.
Well, I mean, I understand the theory, but the point is that within every impacted or challenged community, there will be resistance available, so they won't disappear. And if you look, for example, at comparisons between chickens fed antibiotics and chickens not, it's a much more diverse microbiome in the gut and cecum of the antibiotic-fed chickens. So I think, first off, it's not resource constrained in many of the niches that they live in. Theoretically, you're absolutely right if it were. But the other is that they have the instruments of adaptation within the resistome. So it doesn't take them very long to acquire that through the mechanisms that she just described. But we do have empirical data.
And I just was going to put in a plug for this. We don't support it, but it's a very interesting study. It's called the EnviroChild, because the idea is this is an explicit effort to bring together environmental microbiologists and the clinical world and the microbiome world to really understand how the microbiome forms. And this is in particular case of developing countries, and they're looking at this relationship between diarrheal pathogens and the environment in I think it was Southeast Asia, and I think possibly India or Bangladesh, I don't recall. In Vietnam. So this is, I think, the one explicit example I can think of where they're actually trying to tie the environment literally to disease. And I think I'm going to close there. Yes. Composed of 1,000 species, 3 million genes, and so on. Okay.
Yes, so Lita, one of the things that I found fascinating was your discussion of the environment and the pathogens being in the environment. On the flip side of that, a really interesting thing is that if you look at the genera of the human microbiome, take something like Neisseria. Neisseria gonorrhea is thought to be a dedicated human pathogen. But the genus Neisseria has 7 or 8 members, 7 or 8 phylotypes that are portions of the normal microbiota. So in as much as we have 2,600 phylotypes of, normally occurring bacteria, part of our normal microbiota, many of these pathogens that can either be environmental or dedicated human pathogens can be thought of as spies in a pre-existing conversation that you have, say, with the genus Neisseria.
Oh,
and it's interesting when people who study something like meningitis show all of these virulence factors of meningitis. The question is, does Neisseria lactamica do that? Or how much of that conversation does it do? And that is an area that you know, was
Haven't even
completely
touched.
unknown and
Yeah.
untouched, yeah.
Yeah.